This is a neuron. Neuron is a nerve cell. You can the gap in the center is the nucleus, which has not taken up the GFAP. GFAP is a special stain. Glial fibrillary acidic protein is the full name. Now with that you can see like this. Actually it is this is the structure, and several fibers go in all directions, and there is an axon. You will see that in another picture. Without the stain we will also see that. This is the motor neuron. You can see these branches like this. These are called the dendrites. This is the cell body, this is the nucleus in the center, and these are the dendrites or the branches. The dendrites of this neuron interconnect the dendrites of this neuron. There is a connection between this. So the sensory neuron and the motor neuron are interconnected. That is how when something, suppose you are walking through a field, a thorn pricks your feet. Immediately you withdraw your hand, your feet, no, because of the pain. How does that reach the center so quickly? It is because the withdrawal is done by the new motor. It's a motor activity, whereas the pain sensation is the sensory activity. The pain has reached and the withdrawal had happened almost at the same time, within seconds. So this is so quick. Actually the neurons are very quick in action. And this one, this axon has got, you can see small gaps there, or like you call the nodes. Actually these nodes are called the nodes of Ranvier, after the person who showed that. And these cells are the Schwann cells, and around that there is a myelin sheath. You don't have to go to those details here. Actually you just think that just understand that there is a sensory pathway through a sensory neuronal activity going to your center. From the center there is a motor neuron activity to the periphery, and both these things happen so quick because the sensory neurons and the motor neurons are interconnected, and the neuron has got a structure of a nucleus, a cell body, and several dendrites which interconnect the dendrites of the neighboring neurons. And there is a long axon connected to the sensory organ or the motor organ, as the case may be, and this has some nodes of Ranvier. And here the Schwann, these cells are the Schwann cells, which has got a sheath around it called the myelin sheath, and there is a Schwann cell nucleus which is at the periphery. This is the structure of a neuron. This is a picture to show how the synaptic transmission occurs, so that there are certain synapses, and there is in the resting state how the sodium pump acts, in the depolarization state how it happens, and then repolarization. This goes on happening, and it is this synaptic transmission which produces the neuronal transmission. The synaptic transmission is actually the neuronal, what you call the neuronal. Here you can see how it happens. This is the release function. The first is the release. Then when the whatever is released has to get bound. This is the binding stage. And then there is a transduction, and finally reuptake. Now these are steps in polarization. Now what happens in this process? There is you saw that there is a dendrite and a neuron. Suppose this is neuron 1 and this is the neuron 2. In the neuron 1, at the end of the synaptic end, there are certain vesicle-like structures, and there are certain neurotransmitter transporters. Very nearby a blood vessel is going. No. Now on the neuron 2 there are receptors. Whatever is released by the first neuron is received by the second neuron. This happens in split seconds. It's not a This is how the binding and the transduction and then reuptake into the same happens. So this is what you call a neuronal transmission, and when it is in this action is going on, this is called the action. Here you can see two pictures. One is without cocaine. If an experimental animal is given cocaine and without cocaine, now what is happening at the synaptic end? You can just see that these vesicles are getting released. What are the vesicles containing? Dopamine. It contains dopamine. There is a dopamine transporter, and the dopamines are sent from the first neuronal end. These are the dopamines are the neurotransmitters. They are bound to the other one, and the chemicals produce a potential which is transmitted as an electrical wave. So both chemical as well as electrical reactions happening in the neuron. Suppose the patient is having cocaine, a cocaine addict, or maybe an experimental animal. Then what happens? This cocaine will block the dopamine transport, because the cocaine has got the ability to bind to these things, and the system is not working properly because these are competing with each other, and the cocaine will block the dopamine to function in the normal way. That is how the drugs happen. This is a picture which shows the entire thing in more clear terms. This is one nerve ending, this is the other nerve ending, and here this is the nerve impulse. Nerve impulse is transmitted into an electrical impulse here, no? Now the nerve impulse, actually it comes and it triggers the release of the neurotransmitters into the synaptic gap. This is a synaptic gap, a junction between two. Now these neurotransmitters are liberated here. Then suppose this channel opens to the neurotransmitters attached to the receptor, then this will go on to the transmitted to the other part as an electrical impulse. Here it is a chemical thing, but it is transmitted as an electrical impulse. This is how the nerve impulse is sent to your brain. So a nerve impulse triggers the release of neurotransmitters, and this is what you are experiencing and to which you are reacting. In music also it is the same process which is happening. That is why this is important, not only in a tone prick but also in musical stimulation. This is happening. Now the sending neuron, the sending response and the receiving neuron, this act in a perfect coordinated way, balanced coordinated way.